Before we begin, this episode covers experimental medical procedures performed outside regulatory oversight. We are not endorsing these actions, we're investigating them. Self-administered gene therapy is illegal in many jurisdictions, medically risky, and ethically complex. This is journalism, not advocacy. We present multiple perspectives: supporters, critics, ethicists, scientists. You'll hear arguments on all sides. The goal is understanding, not persuasion. Let's begin. September 2015. A clinic in Bogota, Colombia. A 44-year-old American CEO sits in a medical chair. No FDA approval for what's about to happen. No clinical trial oversight. No long-term safety data. 2 injections. 1 into muscle tissue, 1 intravenous. The first contains AAV, adeno-associated virus, vectors carrying the follistatin gene, designed to increase muscle mass, potentially slow muscle aging. The second contains telomerase, an enzyme that lengthens telomeres, the protective caps on chromosomes that shorten as we age. Her name is Liz Parrish, CEO of Bioviva, a company pursuing anti-aging therapies. And she just made herself the world's first human test subject for an experimental age-reversal gene therapy combination. No regulatory approval, no ethics committee, no safety net. Why would someone do this? Here's what she believed. Aging kills 100,000 people every day, more than all cancers, heart disease, and strokes combined. If aging is a disease, then gene therapy might be a cure. But regulatory pathways take decades. Clinical trials cost billions. She decided, I'll be the proof of concept. My body, my risk, my data. 9 years later, she claims her biological age has decreased. Her telomeres are longer, her muscles stronger. She's still alive. Is she a pioneer or a cautionary tale? This story divides people. Some call her brave, a biohacker challenging a broken system that lets people die waiting for therapies. Others call her reckless, setting dangerous precedents, undermining decades of medical ethics. The truth, as always, is more complicated. This is the story of DIY gene therapy, the regulators it terrifies, the billionaires it excites, and the ethical questions with no easy answers. First, understand what aging is biologically. It's not one thing. It's accumulated damage. Multiple processes, all deteriorating simultaneously. Telomere attrition. Chromosome tips shorten with each cell division. Eventually, cells stop dividing or become senescent. Cellular senescence. Damaged cells that don't die. They secrete inflammatory signals, damage neighboring cells. Mitochondrial dysfunction. Energy production declines. Oxidative stress increases. Protein homeostasis loss. Misfolded proteins accumulate. Alzheimer's, Parkinson's. In 2013, researchers identified 9 hallmarks of aging. In 2023, they updated it to 12. All interconnected. All contributing to what we call getting old. Here's the controversial part. Is aging a disease? The World Health Organization doesn't classify it as one. Most regulatory agencies don't either. But the geroscience movement argues aging is the primary risk factor for nearly every disease that kills us. At 30, your risk of cancer is low. At 70, it's exponentially higher. Why? Because aging damaged your cells. If you could slow aging itself, you'd delay every age-related disease simultaneously. This reframing, aging as treatable condition rather than inevitable fate, drives modern longevity research. And in the 2010s to 2020s, money flooded in. Calico, Google/Alphabet. Billions invested, studying cellular aging. Altos Labs, 2021. $3 billion, founded by Jeff Bezos and Yuri Milner, focusing on cellular reprogramming. Unity Biotechnology, Giro, Juvenescence. Dozens of startups, all pursuing life extension. The anti-aging market is estimated to reach $600 billion by 2025. But regulatory approval is slow. Trials take decades. Some people don't want to wait. Gene therapy. Delivering genetic material into cells to treat or prevent disease. How it works. Take a therapeutic gene, the fix, package it into a delivery vehicle, usually a modified virus, AAV, lentivirus. Inject into patient. Virus infects target cells, delivers gene. Cells produce therapeutic protein. It's not science fiction. It's FDA approved for specific diseases. Lux Turner, 2017. Inherited blindness. Zolgensma, 2019. Spinal muscular atrophy. Multiple cancer therapies using CAR-T cells. But these are for severe, life-threatening diseases. Highly regulated. Extremely expensive. $2 million plus per treatment. Liz Parrish chose 2 targets. Follistatin, a protein that inhibits myostatin, a muscle growth limiter. More follistatin equals more muscle mass, potentially countering age-related muscle loss, sarcopenia. Animal studies showed promise. Human trials? Limited. Telomerase, the enzyme that rebuilds telomeres. Most adult cells don't produce telomerase. Telomeres shorten. Cells age. Turn telomerase back on. Telomeres lengthen. Cells potentially rejuvenate. The catch: cancer cells use telomerase to become immortal. Could activating it cause cancer? Studies in mice suggested benefits might outweigh risks. But human data? Almost none. Why wouldn't the FDA approve this quickly? Because gene therapy can go catastrophically wrong. 1999. Jesse Gelsinger died in a gene therapy trial. Massive immune response. He was 18. 2000. Children in French trial developed leukemia from inserted genes. Regulations exist because bodies are complex. Interventions have unintended effects. Phase 1 trials test safety. Phase 2 tests efficacy. Phase 3 tests at scale. Each phase takes years. To regulators, this is necessary caution. To Liz Parrish, it was unacceptable delay while people age and die. Parrish wasn't alone in her impatience. A growing biohacking movement, people experimenting on themselves outside traditional medicine. 2017. Josiah Zayner injects himself with CRISPR gene editing tool on livestream, trying to enhance muscle growth. 2018. Aaron Treywick injects himself with herpes vaccine, not approved, at a conference. He later died. Unrelated circumstances, but symbolic of risks. Online forums share protocols. Community labs offer gene therapy workshops. DIY CRISPR kits sold online for $200. The philosophy: my body, my choice. Regulations protect corporate interests, not me. The risk: no medical supervision, no quality control, no long-term monitoring. Regulators pushed back. 2019. FDA sends warning letters to companies selling DIY gene therapy kits. 2021. California outlaws selling CRISPR kits for self-administration. 2022. FDA cracks down on clinics offering unapproved stem cell and gene therapies. Their argument? This isn't innovation. It's danger. People will die. Bad data will poison future research. Biohackers counter? Regulatory capture lets people die waiting. Progress demands risk-takers. And beneath the libertarian rhetoric, desperation. People with terminal diseases. No approved treatments. Watching the clock run out. Right to try laws, 2018 in US, allow terminal patients to access experimental drugs. But criteria are narrow. Access is limited. For some, DIY is the only option. For others, like Parrish, it's a choice driven by conviction that aging itself is a terminal condition, just slower. September 2015. Bogota, Colombia. Why Colombia? Because it's legal there. Stem cell and gene therapy clinics operate with less oversight than in the US or EU. Parrish received intramuscular injection of AAV containing follistatin gene, intravenous injection of AAV containing telomerase gene. Dose: not publicly disclosed in detail. Medical supervision: present, but not to FDA clinical trial standards. Follow-up protocol: established through BioViva. Cost: also not fully disclosed, but gene therapy production costs tens of thousands minimum. According to BioViva's published reports and Parrish's claims, 2016, 1 year later, telomere length increased by approximately 20 years' worth, measured by SpectraCell's telomere test. 2019, 4 years later, muscle mass maintained, biological age markers, epigenetic clock, suggested younger profile. No adverse events reported. 2024, 9 years later. Parrish reports continued health, no cancer, sustained benefits. Critical note: these results are self-reported through her company. No independent verification. No peer-reviewed publication of full data. The scientific community's response? Skepticism. Issues raised. Sample size of 1. No controls, no replication. Impossible to draw conclusions. Conflict of interest. Subject is CEO of company selling these therapies. Financial incentive to report positive results. Telomere testing reliability. Telomere length tests can be variable. Single measurements aren't definitive. Publication absence. No data in peer-reviewed journals. Only conference presentations and company materials. Placebo effect. Biological markers can fluctuate. Without controls, attributing changes to therapy is speculative. Most scientists' verdict. Interesting anecdote, not evidence. Needs proper trials. After Parrish's self-experiment, BioViva attempted to commercialize. The business model: offer gene therapies for aging in jurisdictions with less oversight. The pitch: Why wait 20 years for FDA approval when you could access therapies now? The obstacles. US and EU regulators made clear. Offering these therapies to Americans would face legal action. Insurance won't cover experimental treatments. Medical tourism for gene therapy is legally and ethically grey. The outcome. BioViva pivoted, now focusing on advocacy, research partnerships, and waiting for regulatory landscapes to shift. Parrish remains CEO, still advocates for accelerated aging research pathways. And others followed similar paths, with mixed results. Brian Hanley, 2017. Microbiologist injected himself with self-designed gene therapy. No serious adverse events reported, but no dramatic benefits either. Countless unreported cases. Online forums suggest dozens to hundreds have attempted DIY gene therapies. Most don't publicize results. Known risks that materialized elsewhere: immune reactions, inflammatory responses, unintended insertions, genes integrating in wrong locations. No long-term cancer risk data. The survivorship bias question. We hear about successful cases. How many failed and stay silent? Regulators adapted. FDA, 2019 to present. Enhanced monitoring of clinics offering unapproved biologics. Faster enforcement. EMA, European Medicines Agency. Similar crackdowns in Europe. WHO released guidelines on human genome editing and gene therapy ethics. But regulation is national. Gene therapy tourism continues in countries with less oversight. The cat is out of the bag. The question is how to manage it. Let's separate hype from science. What we know about telomeres. Telomeres shorten with age, generally true. Very short telomeres associated with cellular senescence, established. Telomerase can lengthen telomeres in cells, demonstrated. What we don't know. Whether lengthening telomeres extends human lifespan, unproven. Whether benefits outweigh cancer risk in humans, unknown. Optimal dosing and timing, no clinical data. Evidence from research. Mice with enhanced telomerase live longer and healthier, multiple studies, but mice aren't humans. Metabolic rates differ, cancer susceptibility differs. Human trials of telomerase activation. In progress for specific diseases, results pending. Follistatin and myostatin inhibition. The theory. Blocking myostatin lets muscles grow larger, potentially countering age-related muscle loss. Evidence. Myostatin-deficient animals, genetic mutations, have dramatically increased muscle mass. Follistatin overexpression in animal models shows similar effects. Human trials. Exist for muscle-wasting diseases, muscular dystrophy. Results mixed. Some benefit, but not miraculous. The leap. Using it for general anti-aging in healthy adults? No good data. The risks. Heart is a muscle. Could myostatin inhibition affect cardiac tissue unpredictably? Meanwhile, mainstream science pursues aging through regulated pathways. Senolytics. Drugs that clear senescent cells. Clinical trials underway. Metformin trials. Testing if diabetes drug extends lifespan in non-diabetics. NAD+ boosters. Supplements to restore cellular energy. Mixed evidence. Cellular reprogramming. Yamanaka factors to reset cellular age. Altos Labs focus. Realistic timeline. 2025 to 2030. First senolytics might gain approval for specific conditions. 2030 to 2040. Possible broader anti-aging interventions if trials succeed. 2040 plus. Gene therapies for aging, if shown safe. The gap between Parrish's timeline, now, and regulatory science, decades, is the source of tension. The argument in favor of DIY gene therapy. 1. Bodily autonomy. My body, my choice. If I'm informed of risks and willing to accept them, that's my right. 2. Regulatory failure. The FDA process is broken. It takes 15 plus years and billions of dollars. People die waiting. 3. Innovation necessity. Progress requires risk-takers. First heart transplant was called reckless. Now it's routine. 4. Terminal desperation. For someone dying in months, waiting for Phase 3 trial completion isn't an option. 5. Cost reduction. Regulatory requirements make therapies cost millions. DIY democratizes access. Supporters point to history. Anesthesia, X-rays, vaccines, all initially controversial, tested on self-experimenters. The argument against. 1. Safety precedent. Medical ethics exist because bodies are complex. Skipping trials kills people. See Jesse Gelsinger. 2. Bad data poisons research. One reckless case with ambiguous results sets back legitimate research. Regulators become more cautious. 3. Exploitation risk. DIY opens door for predatory clinics selling false hope to desperate people. 4. Informed consent impossibility. True informed consent requires data we don't have. You can't consent to unknown risks. 5. Societal cost. If something goes wrong, public healthcare systems bear the cost of treating complications. Critics point to thalidomide disaster, contaminated blood products, unregulated stem cell clinic victims paralyzed by injections. Some propose middle paths. Accelerated pathways. Faster FDA approval for aging therapies specifically. Right to try expansion. Broaden access to terminal patients and those with limited options. Regulatory sandboxes. Jurisdictions where experimentation is legal under monitoring. International coordination. Harmonize standards so treatments approved in one developed nation are recognized elsewhere. But implementing these requires political will. And aging isn't seen as urgent by policymakers. Meanwhile, people age, and some don't want to wait. 2024, 9 years post-injection. Liz Parrish's status: alive. No reported cancer. No catastrophic adverse events. Still CEO of BioViva. Still advocating. Claims continued positive markers. Maintained muscle mass. Cognitive sharpness. Biological age measures show younger profile than chronological age. But remember, this is self-reported. Company-funded testing. No independent verification. Does this prove gene therapy worked? No. Does it prove it's safe? Absolutely not. Does it prove she's lying? Also no. What it proves? In at least one case, these interventions didn't cause immediate catastrophe. What we still don't know: did the therapy work? Telomere length fluctuates, without controls. Attributing changes to therapy versus natural variation is impossible. Is she healthier than she'd otherwise be? No counterfactual. No parallel universe where she didn't get the therapy. Will there be delayed effects? Cancer can take decades to develop. Cardiovascular issues, autoimmune reactions. It's been 9 years, not 30. Would it work for others? Genetic variation means responses differ. Sample size of 1 tells us almost nothing. The scientific consensus. Interesting case study, not evidence. Proves nothing, except that someone was willing to try. The broader field isn't waiting for Liz Parrish's data. Current aging research frontiers. Partial cellular reprogramming. Temporarily activating Yamanaka factors to reset cellular age without causing cancer. Altos Labs bet. Senolytic drugs. Unity Biotechnology and others in Phase 2 trials. Could clear zombie cells. Gene therapies, regulated. Targeting specific diseases of aging. Alzheimer's, Parkinson's, heart disease. AI-designed interventions. Using machine learning to identify drug combinations that slow aging. Money continues to flow. Billions. The question isn't whether anti-aging research happens. It's how fast, how safely, who controls it. And a question Parrish's case highlights: who gets access? If anti-aging therapies work, will they cost $2 million like current gene therapies? If so, only billionaires afford them. Longevity becomes another axis of inequality. Or could scale and competition drive costs down, like happened with HIV antiretrovirals? The DIY movement argues regulatory barriers keep prices high. Open access accelerates democratization. Regulators argue cutting corners makes therapy dangerous for everyone. No one has solved this tension yet. So where does Liz Parrish's experiment leave us? She didn't prove gene therapy for aging works. She didn't prove it's safe. But she did prove someone was willing to be the guinea pig, and that she survived, at least so far. In 10 years, maybe 20, we'll have real data from proper trials. Maybe we'll look back at Parrish as a visionary who was right, but early. Maybe we'll see her as reckless, who got lucky and could have caused a regulatory backlash that delayed real progress. Or maybe, most likely, it's both. Complicated. Human. Neither purely hero nor villain. Science advances through careful experimentation, but also through impatient rebels who refuse to wait. History remembers both. September 2015. A woman injects herself with experimental gene therapy. No approval, no oversight. Betting her life on an idea. 9 years later, she's still here, making the same arguments, still dividing opinion. Was she right to do it? That depends on your framework. If you value individual liberty above all, yes, her body, her choice. If you value collective safety and rigorous science, no, reckless and irresponsible. If you believe aging is a disease killing 100,000 people daily, maybe desperate times justify bold action. If you believe medical ethics exist to protect us from our own desperation, maybe those protections matter more. We didn't make this episode to tell you what to think. We made it to show you the complexity. Liz Parrish's experiment is happening in real time. The outcome isn't known. She might live to 120. She might develop cancer at 65. We don't know. What we do know, aging research is accelerating. Gene therapy is becoming more sophisticated. Billions are being invested. The question isn't whether we'll try to extend human lifespan. We already are. The question is, how? How fast? How safely? And who decides? Liz Parrish decided she wouldn't wait for permission. History will judge whether that was brave or foolish. This is Cosmic Canvas, where we explore science at its most controversial edges. Until next time, ask hard questions, demand good evidence, and remember, just because something is possible doesn't always mean it's wise.